Thank you, Liz. Good morning, good afternoon. Before we get to our invited speaker, I'll give you a brief introduction of some of the key HiFi applications being used for repeat expansion disease research. At PacBio, we believe in offering our customers the best technology for the application, whether that be long or short read sequencing. With HiFi sequencing, we offer highly accurate long reads, enabling you to access more of the genome, resolve dark regions like repeat expansions, and do full-length RNA sequencing, which is critical for disease research, as we'll see today. Our SBB sequencing technology offers high accuracy short reads, which is best for needle in a haystack applications where finding low frequency variants is essential. Our current HiFi sequencer is the Revio system, and we continue to release new software features and applications. Most relevant today are the Kinnex kits for full-length RNA sequencing and the new PureTarget repeat expansion panel. Our new high-throughput library prep kits, the HiFi Prep and HiFi Plex kits, might be of interest to folks wanting automated whole genome workflows at a lower price point. Let's begin with PureTarget. The PureTarget repeat expansion panel is part of an end-to-end PacBio solution for comprehensive characterization of repeat expansions. The workflow starts with high-quality DNA extracted with nanobind, panDNA, or CBB kits. The PureTarget repeat expansion panel has all the reagents you need for target enrichment and library prep for a panel of 20 repeat expansion targets relevant to clinical research. For sample barcoding, you can use the SmartBell adapter index plates. PureTarget libraries are optimized for sequencing on Revio and Sequel IIe, and we have supported repeat genotyping software in SmartLink version 13.1, which is running the recently published Tandem Repeat Genotyping Tool, or Target, under the hood. Here are the 20 targets included in the PureTarget repeat expansion panel. On the leftmost column are the relevant diseases with prevalence in the middle, and on the right are a list of the genes. The panel includes genes for ataxias, including the very challenging FXN and RFC1 genes associated with Friedreich's ataxia and Canvas disease, respectively. We include FMR1 for Fragile X and HTT for Huntington's disease, as well as the myotonic dystrophy genes DMPK and CNBP. Today's webinar will focus on C9orf72, which is the most common genetic cause of hereditary ALS and FTD. This target is extremely challenging to genotype due to long and highly variable stretches of GC-rich sequence. And as the product owner of PureTarget, I'm really excited to see how this kit will accelerate C9 research. There are a variety of different targeted sequencing methods out there, like PCR amplicons and hybrid capture. But for repeat expansions, the PureTarget method is the most accurate. PureTarget uses the CRISPR-Cas9 system and guide RNAs to target the repeats, cutting the DNA on either side of the repeat. We then ligate SmartBell adapters to the cut ends and digest away the non-SmartBell templates, leaving an enriched library for the repeat expansion targets. Compared to other methods for target enrichment that use PCR, like PCR amplicons or hybrid capture, PureTarget libraries have several advantages. They retain methylation, they do not suffer from GC dropout, they have less size bias, which means more reads for long repeat expansions, and there are no replication errors stemming from the use of PCR. While the PureTarget product uses the same core method as our previous NoAmp product, it is a reimagined, robust, and scalable targeted sequencing workflow. With a faster library prep time of 8 hours, optimized sequencing conditions, and repeat expansion genotyping in SmartLink 13.1, you can go from sample to answer in 3 days. This workflow is about half as long as long-read hybrid capture workflows and conveniently has no DNA shearing or size selection. The DNA input requirements are 1 to 4 micrograms of high molecular weight DNA, and the resulting barcoded SmartBell libraries can be multiplexed. You can run 24 samples per Sequel IIe SMRT Cell and 48 samples per Revio SMRT Cell. The supported sample types for PureTarget are human blood and cell lines extracted with nanobind. We have a new human whole blood extraction protocol using RBC lysis in the pan-DNA kit. And for cell lines, you can use either pan-DNA or CBB kits. We're currently running a promo for a 25% discount off the Nanobind pan-DNA kit and the PureTarget kit when purchased together. On the right is a comparison between a Nanobind extracted sample and the same sample extracted with a third-party kit. This sample has a large CNBP expansion. This comparison illustrates that we get higher coverage, especially for expanded alleles with high-quality Nanobind DNA samples. For the Nanobind sample, we have 42 reads spanning the expanded allele compared to 16 reads for the other extraction kit. These samples were processed with 2 micrograms of starting DNA and sequenced in an 8-plex. Note the expansion is highly mosaic with variation in the length of the repeat, and we obtain reads up to 19 kb for the Nanobind sample. You can learn more about PureTarget by visiting our website, where we have an application note that contains helpful information about sample types and extraction methods. And we also have 4 data sets on PacBio.com that include a set of Coriell reference samples with known expansions, as well as an RFC1 data set from one of our collaborators who is a clinical researcher. Now let's switch gears and talk about Kinnex kits for RNA sequencing. The Kinnex kits are PacBio's latest offering for scalable, cost-effective RNA sequencing. These kits are based on the MAS-seq method for concatenating smaller amplicons, such as cDNA and 16S rRNA, into longer fragments. This takes advantage of the HiFi read length to increase throughput. For example, using Kinnex single-cell RNA kit, one can expect to get 80 to 100 million single-cell cDNA molecules on one Revio SMRT cell, which is sufficient for single-cell RNA sequencing without the need for orthogonal sequencing with short reads. The Kinnex full-length RNA kit concatenates full-length cDNA generated using the IsoSeq Express 2.0 kit. By concatenating cDNA into larger fragments for HiFi sequencing, the throughput is increased by 8-fold to an expected yield of 30 to 40 million cDNA molecules on one Revio SMRT cell. Samples can be multiplexed by different barcoded cDNAs in the IsoSeq Express 2.0 kit, with further multiplexing capacity enabled by using barcoded Kinnex adapters. For analysis, the updated IsoSeq workflow, starting with SMRTlink 13.1, generates isoform classification and abundance information, which can be plugged into tertiary tools for quantification and differential analysis. To give you an example of the throughput increase using Kinnex on Revio, we sequence the same universal human reference RNA with and without Kinnex. Without Kinnex on Sequel II system, we obtained 3.2 million reads, compared to 20.5 million reads with Kinnex on Sequel II. Then compare that to 47.3 million reads with Kinnex on Revio. This represents a 16-fold throughput increase. Further, we showed that the concatenation does not skew transcript distribution, nor does it change isoform abundances. With Kinnex, you just get more reads with the same accuracy. For most studies, such as isoform discovery and comparison of moderately expressed transcripts, we recommend multiplexing 4 samples per Kinnex library with a target yield of 10 million reads per sample. Based on US list price with our kits and sequencing consumables, this comes to $488 per sample. For large cohort studies, we can further reduce sequencing depth to 5 million reads per sample, allowing for a multiplexing of 8 samples per Kinnex library, lowering the cost further to $304 per sample. With that, I want to thank you for your attention, and I'll turn it over to our invited speaker, Evan.
So I'll go ahead and get started. Hey everyone, my name is Evan Udine, and I am in the 4th year of graduate school at the Mayo Clinic in Jacksonville, Florida, working in Dr. Marka Van Blitterswijk's laboratory. Today I'll be talking to you about some of my thesis work and some of the other great work going on in our lab. First, I'd like to thank PacBio, especially Liz and Sarah, for inviting me to present today. where I'm going to talk really about our work using long-read sequencing to understand and characterize C9orf72 repeat expansions and transcripts using HiFi sequencing. So just as an overview, I'll go into a little bit of background on the diseases that we're studying and on C9orf72 itself. Then I'll go into our efforts to actually sequence through the C9orf72 repeat expansion, and end with some brief overview on our transcriptomic studies around C9. So just to start with some background, the two diseases that our lab primarily focuses on are ALS, or amyotrophic lateral sclerosis, and FTLD, or frontotemporal lobar degeneration. So ALS is the most common adult onset motor neuron disease caused by degeneration of both upper and lower motor neurons. Typically with ALS, you see an age of onset between 40 and 60 years old with a survival time between 2 and 5 years. And in general, about 90% of the cases of ALS are sporadic and 10% are familial. FTLD, on the other hand, actually represents a pathological diagnosis that's actually given upon autopsy, but is clinically most associated with frontotemporal dementia. And FTLD is the second most common cause of dementia under the age of 65. And typically you see an age of onset between 50 and 70 years old with a survival time of 6 to 11 years. And overall, about 40% of cases have a positive family history, with 10% of those exhibiting autosomal dominant inheritance pattern. So as the title of this slide and the previous slide suggests, we consider ALS and FTLD to exist as part of a disease spectrum. We say that for a few reasons. One is because there's a substantial overlap in diagnosis, but also in pathology and the genetics. So here I just want to highlight two of the most relevant ways these overlap. The first of which is in the pathology, which is TDP43, and that's present in about 95% of ALS cases and about 50% of FTLD cases. So TDP43 is a nuclear localized RNA binding protein that plays a role in regulating splicing and suppressing cryptic exons. And under pathological conditions, it mislocalizes from the nucleus to the cytoplasm, as you can see in these images on the right. So the other major way that these diseases overlap is they share a common, or at least the most frequently observed genetic cause, which is a repeat expansion in the gene C9orf72. So specifically, this is a non-coding hexanucleotide repeat expansion of the sequence GGGGCC, where in healthy individuals, you typically observe between 2 and 30 copies of this repeat, whereas in affected individuals, you actually see upwards of hundreds to thousands of copies of the repeat. And this is potentially pathogenic in three main ways. The first is through loss of expression of the gene via haploinsufficiency. The second two are gain of function mechanisms. So you can get the inclusion of these small RNA foci that are present in the nucleus and made up completely of the repetitive RNA. And these actually sequester other RNA binding proteins and disrupt normal cellular processes. The other are through dipeptide repeat proteins, and these are proteins that are produced directly from the repetitive RNA itself via a process called repeat-associated non-AUG translation, or RAN translation. And among the transcripts of C9, there's three primary transcript variants that have been well studied and well characterized by our lab and by others, though there are others annotated as well. These are the three that I'm going to focus on in the later stages of this talk. And importantly, cases with C9orf72 repeat expansions will likely display TDP43 pathology as well. So for us today, we're really focused on C9orf72 and how we're characterizing this repeat expansion, which in the past has primarily been studied using southern blotting, which I would say is currently the gold standard for studying repeat length. You can see an example of a southern blot on the right, where at the bottom of this gel, you can see this really dark band representing the wild type allele. And up at the top of the gel, you can see the expanded allele, which tends to present in these more kind of smear patterns. So we and others have completed many studies using southern blotting that have revealed much about the length of these expansions, including that the length of the expansion varies between regions and tissues, as you can see in this bar plot down below. You can see in the frontal cortex in red that the expansions tend to be really long in this region, versus the cerebellum, which is in green here, the expansions tend to be much shorter. But in addition to southern blotting, studies, various techniques have also been used to describe other features of the expansion. So for example, we know that the promoter of C9 is methylated, and hypermethylation of this expansion is associated with reduced expression of transcript levels. But other studies have also revealed that the expansion itself is methylated. In addition, the sequence purity within the expansion has not been thoroughly evaluated based on current methods, and the transcriptomic consequences of having a C9orf72 repeat expansion remain uncertain. So our solution here to really kind of overcome some of these challenges is to use long-read sequencing. And I just want to highlight here why long-read sequencing, which can be defined as any technology that provides single molecule reads up to and even now exceeding multiple kilobases in length. And at the DNA level, some of these advantages are that it can be used to cover complex, highly repetitive regions of the genome, specifically to identify structural variation, detect novel repeat expansions, and fill in missing gaps left by short-read sequencing studies. And at the RNA level, we think about it, the advantages being that we can sequence entire transcript variants, we can span splice junctions in all of our full-length reads, and we can actually integrate it with existing short-read sequencing datasets to improve the quantification of both annotated and novel transcripts. So I'll spend the majority of the time today on our studies actually sequencing C9orf72, where our primary goal here is really to determine the repeat length, the purity, and the methylation levels of the C9 expansion itself. And the way that we've been doing this recently or the last couple of years is by using PacBio's no-amp or no-amplification sequencing, which to provide an overview allows us to sequence a specific targeted region of the genome in genomic non-amplified DNA, which is really important in this context as C9 repeat expansions can't really be amplified as of now due to its GC richness and the size. So the way that we do this is we use CRISPR-Cas9 to target a specific region of the genome, which in this case is the C9 repeat expansion and a small flanking region on each side of that, which we then cut out and pull down for sequencing. So our initial analyses of the repeat expansion to determine the length and the purity were completed with repeat analysis tools, and then we called methylation using CpG tools and PB-Jasmine, and then perform further downstream analysis using custom R and Python scripts that we'll also use for a lot of the visualizations you'll see today. And before going further, I do want to note also that for the no-amp studies that I'm going to highlight today, the samples were not multiplex, so we included just one sample per SMRT cell, and they were sequenced on the Sequel II. So our lab has published one study using no-amp sequencing. This is a few years ago now. In this study, we used DNA extracted from the cerebellum of 28 individuals, which, as you may remember from the intro, is a region that tends to have relatively shorter repeat expansions. But importantly, what we found here is that the length measurements that we obtained from the no-amp sequencing data correlated significantly with the estimated length from our southern blotting results that we had previously generated. And notably, we also noted that shorter C9 repeat expansions tended to be associated with a longer survival time and an increased burden of dipeptide repeat proteins. And while we do have plans to extend these studies to more samples and more brain regions, our most recent work has been focused on studying these repeat expansions in blood. So specifically here, we've highlighted some cases that appeared to be interesting based on previous southern blotting studies. In total here, we completed no-amp sequencing on 34 samples, where we kind of have divided the analysis into two main strategies. The first is evaluating the length of the expansion, and for that, we used a little bit less stringent criteria, where we include one full sequencing pass and a predicted accuracy of 0.8. And for the purity and the methylation analysis that I'll talk about a little bit later, we're using a more stringent criteria where we require 3 full sequencing passes and an accuracy of 0.99. So just starting with the length analysis, I first wanted to just look at the number of reads here that fully cover the expanded allele for C9. Each one of these bars is a unique sample, and this is just the number of reads that we had covering the expansion. And overall, about 70% of samples had at least 10 reads spanning the full expansion. And you can even see a couple samples here had greater than 200 reads that captured the expanded allele. We also wanted to look at the distribution of the lengths that we were capturing. So this is just a histogram plotting the number of repeats across all samples kind of combined into one plot. What you can see here is on average, the median number of repeats that we detected was about 1,400. With some being over 4,000, which would correspond to a read length or an expansion length of 24 kilobases long. And one thing to kind of point out here is that we kind of see two peaks here. And this first peak actually does represent an expansion, even though it looks quite a bit smaller than the rest. This is actually really driven by one sample that I will show a little bit more closely in a few slides. So how we initially visualize this data is through waterfall plots, which we can see here on the left for one representative sample. And this plot shows each unique read for this region for this one sample stacked on top of each other on the y-axis. And on the x-axis, we see the position, or essentially the read length. And we can really clearly distinguish the wild type allele, which is shown on the top of the plot here, which has about 80 reads for the sample, and about 40 reads down below that correspond to the expanded allele. And then we color those reads by the actual repeat motif that we expect. So in this case, like we said in the intro, the expected motif would be GGGGCC. So that's colored here in blue. In gray are actually alternate sequences. So as you can see, there seem to be quite a bit of interruptions to this sequence, but keep in mind that this is our length analysis. So for kind of further studies of the purity and potential interruptions, we can wait a few slides in order to look at this motif as well as some alternative motifs. The other thing that you'll notice here is that there appears to be quite a bit of variation to the length of the expansion, even within this one sample. And that's something that we observe pretty consistently across samples, so that the way that we're assigning a length to a sample at this point is by using the 90th percentile estimate of all expanded reads. And this is because we previously observed a slight bias to capture more of the smaller reads than the longest reads. And when using that measurement, you can see here on the right in this scatter plot that the number of repeats that we detected with NOAMP sequencing in the blood, again, significantly correlates with the number of repeats detected using Southern blotting. And I also want to note, even though I don't show it here today, that in general, we also will always look at multiple length measures. So not only will we use this 90th percentile measure, but we're also looking at things like the median, the mean, and the maximum as well. And in general, we tend to see kind of similar relationships between our NOAMP data. So one of the unique things about this cohort of blood that we sequenced is that we were able to include blood from multiple time points for 6 samples. Therefore, we could evaluate the length of the expansion over time. And what we observe is shown here on the right for both NOAMP sequencing, which is indicated by the solid line, and Southern blotting, which is indicated by the dashed line. So for most of these samples, you can see that the expansion tends to be quite stable over time with not significant changes to the length across multiple years in some cases. However, in some samples, kind of most notably in the blue sample, you can see that the length tends to be quite dynamic. And that's reflected very well in both our NOAMP sequencing data and Southern blotting data. So kind of confirming what we've previously observed with the Southerns. The cohort also included multiple families, and I want to highlight one of these here, which in our previous Southern blotting study, we reported an incidence of a paternally inherited contraction, where the expansion becomes smaller in the offspring compared to the parent. And we actually observe frequent contractions when the expansion is inherited from the paternal parent in that previous study. So here at the top left, we have a pedigree for one example family. You can see the father here was affected with ALS, had an age of onset at 58 and an age at death of 62, and the 2 offspring that we also obtained NOAMP sequencing data for. And at the time that we collected the blood from these individuals, they were not affected with the disease. So to the right, you can see the corresponding waterfall plots for all 3 of these samples, and down below the estimated number of repeats for each sample. And what you can see is that we did confirm the contraction that was inherited from the paternal parent. So you can see in the father, the repeat length was about 1,400 repeats long if you take the 90th percentile. In the first offspring, the repeat length was about 80 repeats long. And in the second offspring, that was about 800 to 1,000 repeats long. So these significant contractions. And notably, if you direct your attention here to the waterfall plot in the middle, this is actually that sample that kind of stood out on the histogram. So you can see here we have over 200 reads mapping to the expanded allele, and it tends to be much, much smaller, which is why it kind of had its own peak on that histogram. So we also wanted to look at the sequence content of the expansion. So again, I'm just showing a waterfall plot here, this time, again, removing the wild type allele, zooming in really on the expansion. And we also used the more stringent criteria for this analysis. And now instead of just coloring the expected GGGGCC motif in blue, we're actually coloring alternate motifs that we frequently observe to be interspersed throughout the expansion. And you can see that these other motifs tend to be small deletions or substitutions to the expected GC sequence. And you can see that there's very little gray, which means that basically all of the interruptions that we observe are actually still composed of GCs. So when we quantify this across all the samples in this study, approximately 80% of the reads tend to be purely of the GG-GG-CC repeat. And there's a little bit of variation between samples and even within samples. But when you look at the total GC content within these samples, it tends to be about 99% GC rich. And we've also completed some preliminary methylation analysis. I really want to emphasize that this is preliminary here, as we're still working on the best way to proceed for us. But as of now, we've created these kind of waterfall-like plots for methylation. So what you can see here on the x-axis is actually CPG position. So this is not all base pairs within the repeat itself. It's actually just the predicted CPGs. And then each read, again, is stacked on top of each other like the waterfall plot. And then the color represents the predicted methylation score. So anything in bright red is predicted to be highly methylated or have a high probability of methylation, and anything in white tends to have a low probability of methylation. And since we're capturing the full flanking region in these reads as well as the expansion itself, the expansion is kind of composed of the middle portion of these reads, and you can see, in this case at least, the expansion appears to be very highly methylated. But this is just one representative sample, and we're really continuing to optimize this analysis, hoping to include kind of the newer tools like Target and TarViz into this analysis as well. So that kind of wraps up our NOAMP blood study, at least for this presentation. And now I'm going to switch gears a little bit to a pilot study that we completed recently with PacBio and the Genome Analysis Core up in Rochester at Mayo Clinic, testing the new PureTarget technology. So we did this test on the Sequel II before we were able to do it on the Revio, and we included a total of 16 of our own samples. So we designed it in a way that we could test the multiplexing as well as a one-plex like we did in the NOAMP studies. And we included only samples that had Southern blotting and NOAMP available. And again, this was analyzed now with Target and TarViz, kind of those updated tools that have become available. And just to see the whole panel here on the right, you can see that this time we're not actually just capturing C9orf72, but we're actually capturing 20 targets that are known to harbor these repeat expansions. So I first wanted to just highlight the overall coverage that we obtained for one of the samples. And you can see for each of the targeted regions, we observed at least 600 reads to each target, and over 1,000 reads to many of the targets. And specifically looking at C9 for this one sample, we actually had about 750 reads to this region as well, capturing both the wild type and the expanded allele. Obviously, since we're primarily interested in C9, we did want to look at the coverage of the expanded allele, comparing the NOAMP data to the PureTarget coverage. So you can see that here in orange, the PureTarget, and in blue, the NOAMP number of reads. For this first SMART cell, which was a multiplex of our 14 blood samples, plus 2 PacBio controls, we actually achieved greater coverage in the PureTarget analysis for 10 out of the 14 samples. But then when you compare the two one-plexes, so the non-multiplex samples, this is more of a direct comparison to what we did in the NOAMP sequencing data. We actually had about a 4-fold increase in the number of repeats, or the number of reads rather, for the second SMART cell, and a similar increase for the third SMART cell. The second SMART cell here including one blood sample, and the third SMART cell here including one brain sample, which I'll talk about separately in a couple slides. So of course, we also wanted to look at the expansion lengths that we were able to detect in the PureTarget data. So to highlight how we did this, we initially called methylation sites using PB-Jasmine, again, used Target and TarViz for the analysis, and then plotted the results with our custom R and Python scripts. So you can see from the distribution of read lengths here that overall the average distribution was relatively similar to what we obtained in our previous NOAMP sequencing studies. However, the maximum length here is greater than what we could previously obtain. So the longest read here was nearly 40 kilobases long, which is kind of far better than what we achieved with NOAMP. And we actually had multiple reads here exceeding 30 kilobases long. So this was great for us to see. One of those reasons we were really excited to see that about the expansion length is based on what we know about some of these samples in the Southern blotting data. So this is the one sample that was a brain sample that we had previously generated NOAMP on. And you can see that we only had a few reads in NOAMP sequencing data, and the maximum tended to be about 15 kb. In the PureTarget test, again, just with a one-plex, we increased the number of reads by 4 to 5-fold, and actually got much longer reads. So the longest read was nearly 30 kb in length. And this actually, what we see in PureTarget is really what we would have expected to see based on the southern blotting data that we have on this sample. So that was great to see. So that covers most of the progress that we've completed thus far in terms of sequencing C9orf72. Just to highlight some of the next steps, we'll be completing association analyses with various clinical and pathological measures. Our preliminary analyses suggest that there's some associations between repeat length and C9orf72 transcript levels. And we'll also be measuring how the expansion purity and methylation changes over time in both the longitudinal and familial samples, like we did with the expansion length. So one of the things I alluded to about that brain sample is that it's been difficult to capture the expansion in the NOAMP data in these brain regions that are known to have longer repeat lengths, like the frontal cortex. However, we're hopeful that using PureTarget is going to help us overcome some of these challenges. And we're currently completing a pilot using the PureTarget kit on the Revio at the Mayo Clinic Genome Analysis Core. I was unfortunately unable to get that data generated in time for today, but we will be testing this on multiple sources of DNA, including blood, brain tissue, and some cell lines. And for this, we plan to include approximately 100 cases, 100 samples across multiple regions. So now I'm going to switch gears a little bit again to talk about some of the transcriptomic studies that we're doing on the lab, specifically focusing on C9orf72 again. So the goal here for our transcriptomic studies is to really examine the consequences of having a C9orf72 repeat expansion, where we're specifically aiming to identify novel transcript variants and alternative splicing events in C9 cases compared to non-C9 cases and controls. And I'll clarify now that when I say C9 cases, I mean generally ALS and FTLD cases that are known to have a C9orf72 expansion. When I say non-C9 cases, I mean ALS and FTLD patients that do not have a C9 repeat expansion, and controls being non-neurologically affected controls. So to do this, we're leveraging both short-read and long-read RNA sequencing. And what I'll say for now is for our long-read sequencing studies that we have generated thus far, we've been using PacBio's IsoSeq. And that's been generated for the frontal cortex and a few cerebellar samples, but we do have plans to fully sequence the cerebellum and motor cortex in the future as well. I first just want to highlight a couple of the short-read studies that we've now completed, one being in the frontal cortex that was published back in 2019, and one being in the cerebellum that was just published about a month ago that included nearly 200 samples. And actually in that study, we did include 2 of the cerebellar IsoSeq samples as validation for some of the novel splicing events that we identified. So I want to show 2 of those here. So in our short-read RNA-seq study, we used leafcutter to identify alternative splicing events, both annotated and cryptic. So here I'm just highlighting 2 of those novel cryptic splicing events. One is in the gene STK10 and one is in the gene EFR3A. And without getting into too much of the detail, in STK10, we see this cryptic splice junction to be present about 47% of the time in C9 cases and 10% of the time in controls. In EFR3A, it's a bit more complicated and challenging to see, but you actually have the presence of this cryptic splicing event in about 18% of C9 cases and about 3% of the time in controls. So down below, we plotted these schematic representations showing the coverage in the IsoSeq data for just 2 samples. So in blue, we have 1 control case, and in red, we have 1 C9 case. And for STK10, you can see that all 11 reads in this region for the control individual mapped to the canonical splice junction of exons 14 and 15, whereas in the C9 case, 7 of the reads mapped to the cryptic splice junction in exon 14, and 1 read mapped to the canonical splice junction. Similar for EFR3A, where in the control individual, 7 reads mapped to the canonical splice junction between exons 1 and 2, whereas in the C9 case, you did have 3 reads mapping to the canonical splice junction. You actually had 3 reads on each side of this novel cryptic exon that we identified from our short read study present in the IsoSeq data as well. However, our most complete long read cohort so far for the IsoSeq is for the frontal cortex. We've currently done this on 102 individuals. Just to provide a brief overview of what we've seen so far, though these numbers are pretty consistent across samples, overall we obtained about 1.7 million reads per sample with an average read length of about 2.4 kb or 2400 base pairs long. And I should note here that the way this was designed, we did multiplex 2 samples per SMRT cell, so that's probably why we're seeing about just under 2 million reads per sample. And in terms of the length distributions, you can see it's pretty consistent across samples, though we do have varying number of reads between each sample. Because I'm focusing on C9 today, I just wanted to plot all of the transcript variants that we detected in these 102 samples for this gene. So here we plot each transcript on the y-axis and the length or the genomic position on the x-axis. You can see the introns represented here by the black lines and the exons represented by the boxes, and the direction of the transcript represented by the arrows in the introns. And then we grouped these transcripts by various categories. So first, by structural category, we identified full splice matches up here at the top, incomplete splice matches down below, novel in catalog transcripts just below that, and novel not in catalog transcripts at the bottom. And then these are further colored by various subcategories based on their splicing events, but I'm not going to talk about those too much today. So really the first thing we wanted to do was just look at the total gene expression for C9orf72 across all 102 samples. And what you can see on the right here is that, as we would have expected, the expression of C9 is significantly reduced in the C9 group compared to both controls and non-C9 cases. What we're most interested in by using this data, though, is to look at specific transcript variants. So if you remember back to the intro, I talked about 3 primary transcript variants that have been well studied by our lab and others. So starting here on the left, we can see that we do observe all 3 exact matches to all 3 transcript variants here that we would have expected to see. And when looking at the expression, we can see that variant 2 is in fact the most highly expressed, and variant 1 is the least expressed, which is again in line with what we would have expected from previous studies. However, in many samples, we're not really detecting any of variants 1 and 3, and this is one of the challenges we've encountered with the iso-seq data so far. So kind of to overcome that challenge, we've leveraged our existing short read sequencing data that we've generated. So to do this, we've kind of come up with a modified analysis framework that allows us to include both short and long reads. And we do this using both FLAIR and SCANT3, where we can use splice junction support to quantify real splice junctions and keep real splice junctions that are supported by the short read data in our downstream analysis. And with this, we basically create a long read defined transcriptome. So that way we have our annotated transcripts and our novel transcripts that we identified in the iso-seq data. Then to that long read defined transcriptome, we go back and re-quantify our short read results using a tool called Callisto. And when you compare the long read only analysis to the long and short read analysis, you can see that we identified about 13,000 genes that were shared across the two, and a total of about 50,000 transcripts. And this was done by Isabel Kortz, who's a postdoc in our lab. And I should say that she did this on a small subset of samples, and we're currently working to extend this to the full cohort of 102 samples that we have available. But what we did want to point out was for C9, we see significant improvement in the quantification of both annotated transcripts, known transcripts here, and novel transcripts that we previously saw. So adding the short read data has really helped us improve this quantification. And finally, I'll just talk about some of the future studies that we have planned. These are kind of more ongoing data generation efforts where we're including both bulk and single nuclei studies using the newest technology to generate Kinnex data on the Revio so that we can increase the read yield and actually multiplex some of these samples while increasing the number of reads that we have. So at the bulk level, we actually completed a small pilot where we compared the overall yield of Kinnex on the Revio to iso-seq on the Sequel2. These were done on the same 4 samples. The Kinnex data was in the motor cortex. The iso-seq data was in the frontal cortex, but that shouldn't really have too much of an effect on the number of reads or anything. And you can see that we drastically improved the number of reads that we were able to obtain by using the Kinnex method and the Revio. So that was great to see. And for these bulk studies in the future that we're going to be completing on the cerebellum and the motor cortex, we are definitely planning to use the Revio and Kinnex sequencing. We've also recently proposed some single nuclei studies, and we've completed a pilot with the Genome Analysis Core up in Rochester, where we included 2 frontal cortex samples. And you can see the UMAP plot here on the right for one of those samples where we identified over 6,000, or we obtained over 6,000 nuclei, identifying many of the known cell types that we would expect to see using brain tissue. And for these studies, in the end, we'll be using at least 30 samples per region, including the frontal cortex, the cerebellum, and the motor cortex as well. So just to summarize, long-read DNA sequencing, both NoAMP and PureTarget, has really allowed us to capture and characterize C9orf72 and other repeat expansions. I didn't talk about them today, but we also have interest in various ataxia-related repeat expansions that we've been using NoAMP and are excited to use the PureTarget to study as well. And the long-read RNA sequencing has really provided us with reads that span entire transcript, permitting us to thoroughly characterize expression and splicing changes of both annotated and novel transcripts. So with that, I will just show my references, and I would like to thank everyone who contributed to these projects, Dr. Van Blitterswyk and everyone in the lab, all of our collaborators at Mayo, for this especially those at the Genome Analysis Core who have worked with us on a lot of these pilot studies. And very much thank you to PacBio for the eagerness to include us in many of these pilot studies as well and inviting me to present today. And finally, we always are very grateful to all the patients that we receive specimens from. Of course, it's not possible to do this work without their generous contributions. So thank you all for listening, and I'll be happy to answer any questions that you may have.
All right, we are back. So thanks for watching the recording. We will now address the questions. Please give me some moment to go through. We have lots of them. So maybe the first question is for Sarah. People are asking, like, there's a certain set of genes that are included in the panels. Is there an option to, say, remove genes from the panels or add genes to the panels?
Mm-hmm. Yeah, that's probably the most common question that we get for this product. It's easy to remove genes from the panel. You can actually do that in silico. So in analysis, you pass a bed file of all the targets in the panel. And if you don't want data for any of the targets, you can just remove that line from the bed file, and then the data won't be produced in kind of the aligned BAM files and the downstream reports. If you want to add targets, this is something that is not sort of officially supported by PacBio. We can't necessarily guarantee that every new guide design for a new target will work, but we do have some recommendations for design tools to use and strategies around kind of the size of the captured region. So what I would suggest is reach out to PacBio and your local team, and we can give you kind of specific advice on that. We have added targets to the panel. We are working on a number of different custom designs, so it's definitely possible, but it's something that we haven't sort of broadly rolled out, and it's kind of, we can support you kind of on a case-by-case basis.
Awesome. Well, while I have your attention, Sarah, maybe this is a quick question for you as well. What is the minimum input DNA amount required for PureTarget?
Mm-hmm. Yeah, it works with between 1 and 4 micrograms of DNA. We generally recommend starting with 2, but we have good performance with 1 microgram. The amount of data, the number of reads that you get on target kind of scales with the amount of input DNA that you put in for a given sample quality. But yeah, 1 microgram will work. That's kind of the recommended minimum.
Awesome. Thank you. And here's a question for Evan. Because you mentioned about the transcriptome for C9orf72. People are interested, are there following sequences in the V1, V2, and V3 isoforms of C9orf72 available in a publication?
Yeah, so I think there may be a few publications that have it. I know my PI, Marka Van Blitterswyk, published a paper in 2015 in, I believe it was Acta Neuropathologica, where she studied the 3 transcripts at, she pretty thoroughly characterized them. And there I know for sure the amino acid sequences are available, but I believe also other publications will have it as well.
Awesome. And also, while I have your attention, maybe we'll continue, Evan, on the transcriptome side, is you've also showed other genes that have cryptic splicing when you were showing the isoseq data. What is known or hypothesized about genes that are affected by the C9orf72 repeat expansion?
Yeah, so a lot of the cryptic splicing that we see, we're not necessarily attributing directly to the C9orf72 repeat expansion. What we think is happening is that the other pathologies that are induced by the repeat expansion affect a lot of RNA binding proteins, and therefore the downstream effects of those RNA binding proteins kind of lead to those cryptic splicing events and the inclusion of these cryptic exons.
Awesome. Perhaps also one quick follow-up on the transcript. Do you detect antisense repeat transcript?
Yeah, so not at the moment. And I've looked, so I should, to be honest, I've looked at the IsoSeq a lot more thoroughly than I have at the Kinnex data thus far, just because that's a bit newer to us and just on a few samples. And of the 4 samples that we included in the Kinnex, not all of them were repeat expansion carriers. The thing that we see in the IsoSeq is that we see samples that kind of have retainment of the first intron where the expansion would be located, but we don't always necessarily see the actual repetitive RNA. So we don't actually see the repeat in those transcripts at the moment. But one of the things we need to do is kind of still look at the subreads to see if we're capturing maybe bits and pieces of the expansion that are kind of not fully integrated into the downstream analyses.
Gotcha. One more question for Evan before I come back to Sarah. There's lots of questions. So just so you know, we might not get through all the questions in the next 10 minutes, and you will get a recording of this webinar, and also we will get all the questions. So if your question is not addressed in the next 10 minutes, don't worry, we will follow up with you. One more question for you, Evan, before I go back. Does the PureTarget sequencing also shows high methylation within the repeats in the blood samples brain regions?
Yeah, so far it looks very similar to what we saw in the NOAM sequencing. They didn't do any like formal correlations or anything, but we definitely see it in there as well.
Awesome. So a question for Sarah: could long read be used on FFPE samples to determine repeat length?
I would not recommend that. FFPE samples tend to be really degraded, short fragments, kind of low quality. And in order for the PureTarget Cas9 based method to work, you really need large intact strands of DNA so that you can cut on either side of the repeat. If the fragment starting out doesn't span both cut sites, which tend to be about 5 kb apart, you just won't get any kind of on target reads. So yeah, the supported sample types are blood and cell line from human that are extracted with nanobind extraction kits.
Awesome. Another question for you, Sarah. Is there a plan to work on a PureTarget panel for screenings in mice?
Not at this time. I think there's a lot of research interest. There's a lot of mouse models for repeat expansion disease, something that we may be able to do in the future, but we just launched the product about 2 months ago, so we're really focused on the human samples at this point. Okay. In principle, the method should work on mice. You just would need to kind of redesign the guides against that genome and probably do some optimizations for those sample types.
Gotcha. Perhaps there's a question sort of for me, because it's asking if there's any plans to use Cas9 to look to enrich for transcripts. So that's a question for me. I'd say it's actually a quite interesting thought, and I would say as of now we have been focusing on using more traditional like hybridization probes. So we have customers who have been using IDT, twist, or any kind of hybridization methods can work with our cDNA enrichment. It's quite an interesting thought of suggesting this. I'll definitely think about it. Question for Evan. How do you ensure the observed paternal contraction is not due to the dynamic expression in the offspring?
Yeah, so for that, we would have to have, I guess, multiple measurements from the offspring. Like I showed in the one longitudinal plot, in some cases, it does seem to be a bit variable in over time in specific individuals. So as of now, for the samples that I've shown, we don't have multiple time points for those offspring. So I guess we can't fully rule that out. But what I'll say is we have studied more of these contractions than the one that I've just shown. In NOAMP, we've studied 8 contractions that we see that confirmed results that we also saw in Southern blotting. And in Southern blotting study, there's a paper published in 2020 where we looked at many families. And we saw, I think it was 14 or 16 of those contractions present. So I think the chances that they're all down to expansion, to kind of dynamic expansions is unlikely, but I guess it still does remain possible in some individuals.
I see. Okay, here's one more question for you, Evan. How did you generate the list of possible combinations of GCs, for example, GGGCC or GGGC?
Yeah, so I'll say I didn't specifically generate that list necessarily, but the ones that we are coloring in the colors that weren't blue in that waterfall plot were, I think, just determined based on what we observed more frequently within the expansion. So the initial kind of plot was just GG, GG, CC versus everything else. And then when we started to look at the everything else, we saw a few kind of motifs that came up a bit more frequently than others. So we decided to just color those.
Another question for you, Evan. So you mentioned that you were doing brain, and even within brain, for example, you were doing cerebellum versus motor cortex or blood. Can you just elaborate for both, for the repeat expansion, what's the difference of different tissue types? And then for transcriptome, what's the difference of using whole brain or specific regions of the brain?
Yeah, so for the targeted sequencing for NoAmp and PureTarget, one of the things we've observed in the past is that the length of the expansion differs significantly between regions. So in the frontal cortex and the motor cortex, you tend to see really long repeat expansions. In the cerebellum and the blood, they tend to be a little bit smaller. So that's kind of the rationale for studying multiple regions. We've actually seen that the length of the expansion doesn't always associate with kind of clinical features. So one of the things I showed from the NoAmp study was that in the cerebellum, shorter repeat lengths actually conferred a survival advantage, but that's not the case in every region.
That is something I did not know.
Yeah, and then you asked the second part. For the transcriptome, yeah, so we've done a few studies using short read sequencing that have shown pretty big differences in C9 cases, or big differences when you compare C9 cases versus controls in the frontal cortex. We find different alterations than when you compare C9 cases versus controls in the cerebellum. So of course, there's a few reasons. One is that the cell types that are in there are slightly different. The proportions of cells that are in there are slightly different. And especially in C9 cases, if you're looking at FTLD cases in the frontal cortex, you obviously have a lot of degeneration in those individuals versus the cerebellum, the tissue's much more preserved. So you're looking less at kind of neuronal loss and more so at just the transcriptomic changes within those cells.
Gotcha. You mentioned, so you were talking about different regions of the brain. Was there a particular reason to do blood? Of course, accessibility, I imagine, is one reason.
Yeah, definitely the accessibility is a big reason. Obviously, it's great that we can detect it in blood. It can be done ante-mortem, and we can look at the multiple time points, which is something that's, I think, really of interest to us as well. But yeah, just the fact that we can detect it in blood, I think, was kind of for the biomarker purposes and the genetic testing kind of purposes is why we're looking in blood too.
Awesome. And perhaps I'll take a bigger picture question, is that, it's rare that, you're one of the few people who've done NoAmp and then PureTarget. And then for the RNA side, you've done IsoSeq and now Kinnex. How does it compare in terms of both the library prep, the data quality, and the analysis in this evolution of NoAmp to PureTarget and IsoSeq to Kinnex?
Sure. So starting from, I guess, with PureTarget versus NoAmp, in terms of from the very beginning, the DNA extractions were pretty much the same that we did, except that we required less input material for PureTarget, which is great because that can definitely be a challenge for some tissues in some regions. The library prep, I'm not doing. We're luckily working with the genome analysis core in Rochester to do that. From what I get a sense from them is that it's at least as or even more streamlined now with the PureTarget kit versus NoAmp. I think at least with PacBio releasing it kind of commercially, there's a lot of support available. So that's something that we're, I guess, continuing to work on. And then in terms of the analysis and data, yeah, with the PureTarget, we're definitely seeing a lot more data compared to the NoAmp at the raw level, and that translates to how many reads we're getting in the end. In terms of the analysis, it's overall pretty similar. It's not that much more difficult to look at all 20 targets compared to just one target from like a programming perspective. So I've done everything on the command line, not yet in SmartLink, but it's still just as straightforward, and I think will become even easier too, because the more new tools like Target and Tarviz that are coming out are I think really useful for something like PureTarget.
Gotcha.
And then for the IsoSeq versus Kinnex, I would say with both, there's the size of the data can be a challenge, especially when working with a lot of samples. With the IsoSeq, we're working with over 100 samples already, and with that, we've definitely run into lots of challenges computationally, just with getting enough storage and computing power. We've kind of gotten around a lot of those things with splitting things up by chromosome or even further than that, and kind of subsetting the files as much as we can before we go into the downstream analyses. But with Kinnex, we haven't done it at that kind of scale yet, so we don't really have those challenges yet. I foresee they'll be similar. But again, with the kind of continued, I would say, updating of the kind of IsoSeq and the analysis documentation, it's not any more difficult, or probably equally as well outlined to analyze the Kinnex data versus IsoSeq. It's, I think, really just one more step with the S-reads generation, and yeah.
Thank you so much. We are at time. Thank you so much for all the questions. For those that we're not able to answer, we will follow up over email. Thanks again for attending this webinar, and we'll see you soon.
Thanks, everybody. Have a good one.